This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is TWiV, This Week in Virology. This is episode 448, recorded on June 26th, 2017. This episode of TWiV is sponsored by Promega Corporation. Promega is a leader in providing innovative solutions and technical support to the life science industry. The company's 3,500 products support scientists worldwide to advance their knowledge in genomics, proteomics, cellular analysis, drug discovery, and human identification. Founded in 1978, the company is headquartered in Madison, Wisconsin, with branches in 16 countries and over 50 global distributors. Promega products are used by life scientists who are asking fundamental questions about biological processes, applying their knowledge to diagnose and treat diseases or discover new therapeutics. For more information about Promega, visit promega.com/ASV. Hi, everybody. I'm Vincent Racaniello, and you're listening to the podcast all about viruses. Today, we are coming from Madison, Wisconsin. We are at the annual meeting of the American Society for Virology, and I have the entire TWiV team here with us today. Starting on my left, Dixon Despommier.
Hello, Vincent.
Well,
and everybody else.
Dixon, these are all virologists.
I'm not intimidated by that just because I'm the only non-virologist in this room.
Let me ask you one question.
I feel like I'm right at home.
One question, Dixon. How many different genome types are there?
7.
There you go. Dixon has learned. Thank you. Dixon and I started this many years ago. Do you remember the first live TWiV? How many people were in the audience?
3.
We've been very patient. Also joining us, Rich Condit.
Hi, everybody. How you doing?
Now, what is this?
It's a cheese hat. We're in Wisconsin, and a cheese tie. Ann Palmenberg. Is Ann here? She told me I had to wear. We had to wear
She's
these.
not here, so
are
you can take it off if you'd like.
Right. I'm going to. Don't worry about that.
Also, all the way down there, Alan Dove.
Good to be here.
Welcome. Alan joined TWiV after Dixon and I got it started.
And I have to say, Rich, you look strikingly like Bill Nye.
I just got that message. Yeah. My wife just texted me exactly the same thing.
Well, we do need a Bill Nye for microbiology, right? So, you going to be it, Rich?
Sure, with the cheese tie and the whole thing, sure.
All right, and finally, all the way down there at the end, Kathy Spindler.
Hi, everybody.
Welcome to TWiV, and we have a special guest for all of you today from the University of Florida in Gainesville, Mavis Agbandje-McKenna. Welcome to TWiV.
Hello, good afternoon, everyone.
And glad to have all of you here. This is really exciting. We usually don't have the whole crew here, so we're really grateful that you're here. And we have a great audience, so thank you for coming. And we have T-shirts here for you, which we are going to give out today, but I want to prime the pump so that you don't leave. I'm going to throw the rest out at the end of the show, so don't leave. All right, so here's the first one. One more.
Load and lock.
Load and lock. Let's see how this works. Ooh. Well, that curved. And my arm is shot. That's it. So we have a bunch more here. We'll do it at the very end of the show. So those of you that want it, don't move on. We'll be finishing on time, but we'll throw those out at the very end. Now, we have just 2 brief follow-ups.
Wait a minute.
Weather?
Yeah.
Go for it.
62 degrees. It says it's cloudy. That's not my experience. It looks nice to me. It's been beautiful weather here.
There's a broken cloud layer at about 3,500 feet, apparently. Temperature 16 Celsius.
Pilots speak.
We had to put this curtain up for you, otherwise you wouldn't be able to see us. There'd be too much back glare, but it's a beautiful day out there. All right, Kathy, you have some follow-up, right?
Yes, I do. So those of you who listened to the TWiV 2 weeks ago, where we talked about the paper by Rob Kaleda and Ann Palmenberg analyzing male-female balance at virology meetings, we raised the question about the Venn diagram. And those of you who are here at the meeting, you know that that poster is in the main hallway near the speaker ready room. So the Venn diagram, there's an intersection of people who have attended or been a speaker at all 4 meetings. And those 4 meetings are the ASV, the International Herpes Virus Workshop, the Gordon Conference for Animal Viruses and Cells, and the Positive Strand Virus Symposium. And so to have somebody who would be at the herpes virus meeting and the positive strand virus symposium seemed like that would be mutually exclusive. But there are 5 in that part of the Venn diagram. And we speculated that maybe it included a structural biologist, and Rich thought, oh, Joan Stites. And I remembered somehow in the paper or something, I thought that Anne had been at a herpes virus workshop, but in fact, she's not in that 5 people of the Venn diagram either. But those people in that 5 are some structural biologists and an immunologist. And so here are their names: Rafi Ahmed, Brian Cullen, Ari Hellenius, Felix Ray, and Skip Virgin. So there you are. No women were in that group
All
of
of
5.
the male participants.
All right, we need to fix that, right? All right, I have one quick follow-up before we start talking with Mavis, and this comes from Chris Kandayan, who is our good friend at ASM, who helps us with our productions. And he said, you can play TWiV on Alexa by saying, Alexa, play This Week in Virology on TuneIn. I just want everyone's Alexa to start playing TWiV. It only plays the most current episode, but if you finish it, it will play the previous one.
Now, if somebody is playing this episode of TWiV on their Alexa, when you say that, is it going to be sort of an Inception thing?
Yeah, absolutely. That's what I'm looking
That'll
for.
be
That's
great.
why I included this.
They will end up hearing every episode
Yeah.
of TWiV and nothing else from their
Absolutely.
Well, it occurred to me that if you listen for 800 hours, you could hear the whole series.
Is that how many hours we have?
Binge
At
TWiV.
least, yeah.
Does anybody binge TWiV? Nobody. Yeah, you have? Very good.
Alexa. That would be a long plaque essay.
Better than binging. It's better than House of Cards. I sense a skepticism. All right, fine. All right, this episode's about Mavis. So Mavis, you've heard of TWiV, right?
Yes.
So now you get to tell us your life story, starting from the very beginning. Tell us where you were from and brought up and all that.
Right. So yes, I was born in Nigeria in a little village in Bendel State. Name of the village was Ovara, and it was one of very few people in the village. You know, you could stand in the middle and see both ends, and there was 2 little streets, which as a child I thought was long, of course. But then I actually went back when I was a teenager, and I realized that I could stand in the middle of the town and see both ends, whereas I thought it used to take me hours to get from one end to the other. Anyway, so for the first 11 years of my life, I lived in this little village, and I went to a little village school. Not everybody got to go to school, but I did, and that's mostly because my parents actually left me. My father left me in Nigeria when I was 1. My mother left me when I was 2. They both moved to England, which was very common in those days. I was from a state called Igbo State, and people who could afford it sent their kids to mostly England for education. So my father went there to get his undergraduate degree. So I was left with my grandmother. So I grew up with my grandmother. I thought my grandmother was my mother for the first few years of my life. I didn't actually see my mother until I was 10. That's when they could afford to come back to Nigeria to see me. So I stayed there till I was 11, and at that point, I could tell you about my school. You know, I told the education workshop students the other day that I walked barefoot for the first 11 years of my life. Some of them didn't believe me, but it was true. I did not wear shoes. You know, my clothes were made by a tailor, so that was my life for the first 11 years. But my parents felt that I needed to be better educated, and I needed to come and join them in the UK. So they came back to get me. Actually, it happened, and then the first time they came back, which is when I was 10, my grandmother was so distraught that they left me. So they came all the way to get me. They took me all the way to Lagos, and then I had to go back because she was so devastated. She had raised me all these years. But then they took my half brother. So for those people who don't know, I grew up in a polygamous society where my father was, he had 2 wives. And so so there was 2 of us born around the same time, my brother and myself. He took my mother to England, but left the other wife. So I grew up with my other brother, similar, as if we're siblings. Everybody was fine with it. And so they took my brother when they came that first time, when we were 10. And they saw how well he was doing in the UK, that they said, they needed to come and get me to go back, to go with them to England. So this is, I should tell you, is unusual in that in where I grew up, girls did not necessarily get educated. So my mother, for instance, was one of 4 kids. She had to work to send her brothers to school. She was not allowed to go to school. So it was very, and a lot of people that I grew up with, the girls did not go to school. That's just the way it was. So it was very unusual that my parents wanted to take me to England to get educated. So I was very happy for that. So I moved to England at the age of 11 and did all my sort of high school, middle school there. My undergraduate degree, University of Hertfordshire. And I should also back up a bit and say, I moved to England and there was 5 kids born while I was, in Nigeria. So now I'm the oldest of 5 kids and had to come in and get integrated into this family. So that was a challenge, but it was fine. Because I felt at the time that I didn't really belong to this family. But again, this was I got integrated into it. I had to learn English, so my parents got me a tutor, and so once a week she used to come and teach me how to read and teach me how to write. And at school I had to go to the English for Foreigners class, which was very useful. But I did all of that and did my undergraduate degree. When I was ready, I did a double major, human biology and chemistry, because I couldn't make up my mind, and I liked both of them. And then after that, did a PhD in biophysics. Again, where I could use my chemistry. Actually, I was able to use my chemistry in addition to the biology. I made DNA intercalators because I was interested in anti-cancer drugs. That's where as a graduate student, that's where I met my husband, Rob McKenna, who was a crystallographer in the lab. And he wanted to go to the States for his postdoc. He wanted to go to a big lab to do his postdoc. So he contacted Michael Rossman, and Michael Rossman, so I've got to tell you this story. He said to Michael, I have a wife who I want to bring with me. And Michael said, okay, I could maybe create a position for her. Put her on the phone when they were talking. So talking to Michael, and then I had a very thick London accent. So Michael said, okay, I'm going to hire her. I like talking to her. He hadn't even seen my CV or anything, but because he liked the way I talk, he decided he didn't even talk to Rob again on the phone. He decided to hire me for that, so that was fun. So I followed Rob, totally new field to go work on. So we, started working on viruses. I didn't even do microbiology as an undergraduate student. So just to tell you, I hated viruses, I hated bacteria, didn't want anything to do with it. But then when I got to Purdue, obviously, that's what we did and fell in love with it. And I started as a biochemist, the person who made the viruses in the lab and did all the characterization, but then saw how much fun people doing the crystallography were having. So then I decided that I wanted to do everything. So all the way from making my virus to solving the structure. And one thing I didn't learn, though, was molecular biology, and I do regret that. My lab does it now, but I didn't make any mutants as a postdoc. But anyway, I wanted to be able to do everything myself. So another time we started dabbling in cryo-electron microscopy as well. But not a lot, and that was with Tim Baker. So that's really, my education was, that route. And, from there, I could keep going. From Purdue, we moved back to the UK as a research fellow at the University of Warwick. Carried on working on viruses, then started working on Gemini virus as well as parvovirus and bacteriophages at the time. So really establishing ourselves as single-stranded DNA virus people. We stayed there for a couple of years and we were recruited back to the University of Florida to start working, to build a group in structured biology. So when I first got there, you heard me talk, some of you heard me talk on AAV yesterday. I did not want to work on AAV when I got there because to me it was like a virus. Didn't do anything, doesn't cause disease. Why would you want to work on a virus that didn't cause disease? That was my attitude at the time. But then, and I was dragged kicking and scratching into it. I wanted to work on pathogenic viruses. I collaborated with Colin Parrish to carry on working on canine and feline parvovirus, working on minute virus of mice, and branching into human parvoviruses that were pathogenic at the time. But one of the things that caught our attention with the AAVs was everybody was working on it, but not really, the basic science needed sort of more information. And everybody was saying, oh, this is such a great virus because it doesn't cause disease, it's not immunogenic, right? So this is why it's great. But then we realized that that's not actually true. It is immunogenic, and the reason for that is all the studies have been done in mice, and not in large animals or non-human primates, and nobody really looked at humans and the effect. So we decided we were going to tackle this problem and start figuring out about the antibody response, and I think that's really where we've contributed the most, is tackling that and just trying to, because this is now we realize is a big challenge in the field. And that's what I spoke to you about yesterday, and that's how I got here.
So Mavis, when you were growing up, I guess you didn't think about viruses at all, right?
Not at all. At least not in a good way.
What drew you to science in college or in school?
In school, I love science, just discovery. I liked being able to figure things out. That's why I say science. I hated biology because it was just words and facts to remember, which is why I did chemistry. But yeah, I just like to be able to understand and figure things out. So even back in the village, I always did great in science and high school all the time.
So what's your native language?
So I speak Kwali and Urobo. Those are my two languages.
Can you still no? and have you been back to your village?
The last time I went was in 1988. It was a really long time ago.
What part of the country in Nigeria is your village in?
My village is near Warri. I don't know if you know. It's sort of
is up country, down country?
Down.
Down.
Yeah. So that's the biggest place that it's near. So there's a tragic story why I haven't really been back to Nigeria is that my father actually got killed in Nigeria while he was driving from my village to Warri, and that's just because of the unrest there. He was held up at gunpoint and he got shot, and so I didn't want to go back after I had kids because my kids need me. I can't go back at this point, and unfortunately I haven't been back. I hope to get back and take the kids.
So how do you spell the name of your town? I'm looking here on Google Maps.
So the name of my town is actually Ovara, O-V.
This is what we do.
Right. We Google
Or you could type
We look stuff
Orogun.
up, yeah, and we don't know it.
I found over.
We actually got
Ovara.
criticism for looking it up as we were speaking. And what would you rather have us do? Get it wrong and then have to tell
Or
you
you could
next
say
week we got it
Orogun.
wrong?
That might be. Yeah.
Oh, you're almost on the coast.
O-V-A-R-A. Did you see it?
No, I'm in
It's
Portugal.
in Portugal.
Sorry.
Sometimes we
you-
really
you-
get it wrong.
You guys go ahead. I don't want to take it up with this. I'll figure it out.
Not yet.
I'll figure it out. You guys go ahead.
So Mavis, it's an interesting story of you going to college and then finding science, right?
Yes, so exactly. So actually, I did. I went to college. I was very interested actually going into a biotech company and maybe working on some sort of quality control aspect. And then I met Rob, really. So he's the one who, and I didn't want to be a faculty member, just like most of the people. I really didn't want that. I just wanted a job. But then, Rob dragged me into it. He wanted to be an academic right from the beginning. And that's how I got dragged into science.
Do you really have to drag her? Yeah.
No, she drags me.
No, she drags you. Okay.
So how did you end up in Florida?
In Florida, I was recruited by the dean of the Ken Burns.
Right.
Because he knew of my work on parvoviruses and structure, and he wanted somebody to start doing structures of AAV. I think that was the main reason for bringing us there. So he created a space, and for two of us, provided us the resources to get started.
Yeah, Ken Burns was one of the original AAV
Yeah,
guys.
he's the one who said AAV could be used as a gene therapy.
Right.
And Nick Musicka was there at the time,
Yeah,
right?
exactly. He and Nick. And I told the students when I talked to them the other day that Nick was my mentor. He took me under his wing and said, "You're going to work on AAV." Because it's needed.
Mavis, when you went to the Rossman lab, that's when you first learned some structural biology. You
Yes.
did X-ray crystallography first, is that right?
Yes.
So, looking at your lab photos, a lot of pretty intimidating equipment. It almost looks like a machine shop. So do you have any good stories about things that you did wrong in the beginning?
Oh, things that we did wrong, as in my lab.
Did you break any machines at the time, or?
So let me tell you, we have a lot of machines, but I'm not a machine person. We have an X-ray equipment in our lab. I never touch it. The only thing I do is put crystals in there and collect data. When it's broken, I call Rob. So, and that's the truth. When we go to the synchrotron, it's the same thing. I like putting crystals on and collecting data. I don't like touching machines. I can't fix anything. So,
Okay.
What do you use for a synchrotron? Where do you go?
So we go to either Argonne or we go to Cornell.
Okay.
Yeah.
Yeah, I always wondered with all this equipment if that's an impediment to someone going into structural biology, or if you can always find someone else to fix the equipment.
Yeah, no. I don't think it's an impediment at all, because there's always people. I mean, a lot of it's because Rob likes fixing things. Otherwise, we would have somebody. We would have a facility manager who will fix it.
You fix your own car as well? No.
There are many people who fix it. So no, you don't. There's always somebody who can fix equipment.
Right.
Yeah.
Now, maybe for our listeners, you could sort of quickly distinguish between X-ray crystallography and cryo-EM, which is, I guess you learned that later, right? After you had done crystallography?
Well, I mean, crystallography, you have to purify your protein, and you have to form a crystal, which is just a three-dimensional array of your molecules organized with certain certain rules. There's only so many ways you could organize molecules in a crystal, and that's all known. So you need that you have to have this crystal, which just amplifies your signal when you radiate those crystals with X-rays, and it gives you, what you guys may see as the spotty pattern on an image, and that spotty pattern is just information about the electronic distribution of the protein sample, coming from different what you would call planes that cut those crystals. So that's what you have. And it's missing, you guys would have heard, it's missing information, which is information about the phase, where those spots came from. We have to calculate that or determine that to be able to then get our electron density. With cryo, you don't have to do that. You have, with cryo, you don't have to grow crystals, you purify your proteins, you freeze them, hopefully in just one layer, and then you use electrons to collect the information. In that, there's no missing phase information. That's kind of almost in real space. And then in a cryo-electron microscopy, what you hope, at least what I do, single particle reconstruction, what you hope is that you have enough different observations, different orientations of your molecules to be able to add together to get your three-dimensional image from the 2D So really the biggest difference is at the end of the day, you're going to get an electron density map, which just gives you, the electron configuration around each of your atom. But in one, you're using crystals to amplify your signal. In the other one, you're using just numbers and observations to amplify your signal. At the end of the day, you're going to get an electron density map for from both.
So when you start on a structural biology project, do you pick one or the other initially, or do you say we're going to simultaneously use our X-ray vision and hit it with a freeze ray?
No, you don't. So when I did mostly X-ray crystallography, that was the technique that was going to give me the resolution I needed to be able to see atoms. We couldn't really do that with cryo-electron microscopy. But now, because of the software development, microscopes, and the direct electron detector, we're now able to get to the same level of information. So I would say my lab with viruses, we don't do X-ray crystallography anymore. We do mostly cryo-electron microscopy. Because we could get the same resolution.
So for biological structures like proteins and viruses, do you see cryo eventually replacing crystallography, or are they always going to be complementary? Or
I can't say that. I can't.
Well, you could.
No. I can't say that because I've got crystallographers. So the answer is I think they're going to have their different niches. Right now, we're still limited by size. So, with NMR, if it's too big, you can't do it, with solution NMR. With cryo, if it's too small, you can't do it. So for the smaller proteins, you're going to still need X-ray. And also, there's a lot of development with the, X-ray free electron lasers to look at dynamics. And so you're still going to use X-rays for small crystals there. So I don't see it replacing it. I just see they're going to have different niches.
Does one go faster than the other? And what was your fastest structure solved?
So right now, X-ray crystallography could be really fast. And if you have, so it depends if you have a structure already, and you could do molecular replacement, as it were. Let me back up. X-ray crystallography could be very long because you have to get crystals, right? So if you can get crystals, solving a structure once you get a diffraction data is probably faster for X-ray crystallography if you have a molecular replacement model. But for cryo, as long as you get a protein, also you get your virus that will go, you can make a nice specimen. You can get your structures now, sort of in one day or in a couple of days after you collect data. So it's amazing, but it's very, very sample dependent, right?
So from start to finish, what do you think is your fastest structure solved?
Fastest structures solved. I would still say it would have taken us probably still a month, really, to do it. Yeah, because just data processing. I mean, I've never solved a structure at a synchrotron, say, for instance. Some people do it, but we've never done that. So I would still say it'll take a month.
Does the atomic force microscope play any role in your life?
No, I would like it to. Just because I'm interested in visualizing asymmetry in some of our viruses, and I think most of them do. And I would like to be able to expose viruses to certain conditions that lead to conformational changes or protein domains being extruded, and I can use AFM to visualize. But I haven't been able to get somebody who's really willing to work with me, and we don't have one. So I would like to use it, but we haven't had the opportunity.
Now, for a number of years, people have been talking about the possibility of solving structures computationally. Mm-hmm. And for just as long, people have been failing to solve structures computationally. Is that something where eventually we're going to get to the point, maybe not for viruses, but for proteins, where we can get to structure without having to dip a pipette?
I don't see how you can do that. Obviously, you can predict structures, if that's what you mean. And I think once you have a good database, I think you can get a good model of most proteins. But a model is a model. So I'm one of those people who believes that yes, it's a model. It's going to give you an approximation, but it's not necessarily the real thing. So we know that a conformational difference in an amino acid side chain could lead to large consequences in function.
Right.
So the model will give you, it will sample the conformational space and give you an energy minimized position, but it may not be the position in solution. So, maybe other people believe differently, but in my opinion, I think you still need to,
But the models at least are providing
very
some.
good. Absolutely.
What's the quote? All models are wrong, but some are
useful.
Useful.
Yes.
Yeah, I would say, I would believe that. Yeah.
So Mavis, how should you prepare for, if you want to be a structural biologist? Are there any special qualities, or could anybody? Do you have to be strong in math or chemistry or what?
Yeah, I would say, well, I don't want to scare people by saying you have to be, good in math, but obviously you have to have a strong background in the sciences. You have to, be good in chemistry. I would say math would be good. Physics would be good. I wasn't good at physics. Let me just say that. But you need to be able to learn. I mean, a lot of the students that come into our lab, they're coming into the College of Medicine. They are not expecting to go into a structural biology lab. But then they see what we do and the fact that the structure gives you so much information that you can use, that they are willing to learn. And I think most places where they do structural biology will teach you. So we teach how to solve structures. We teach the math behind it to all our students. So I think if you're willing to learn and you have a background in the sciences, then I think you can do it.
Yeah. So if I remember from one of your talks years ago, I think you bring young students into your lab. And tell us about that.
Yeah. So we bring high school students. We actually have middle school students come in, just see what science does. Everybody wants to be on CSI, of course. So they all come in, and we've had schools visit, middle school visit, where the head teacher will come with all the students and we'll set up the middle of the lab and we'll get them to set up lysozyme crystals, and then we'll get them to look at microscopes, and then we'll take them into the X-ray machine. And they're so excited. So we do that for a science club. But every year we have a program that brings in about 80 to 90 high school students into UF, and they pair them with labs. So we always have one of them that comes in, and we have 6 weeks with them where we teach them how to do science. We bring high school teachers into the lab, same thing. And it's very simple. We try to get them to set up lysozyme crystals and move things around on the graphics, and some of them actually developed syllabuses based on this and take it back to their schools, and they will teach their students.
I think high school teachers is great because for them to see the science, that's pretty rare. Then they can take it back. It's perfect.
They take it back, and we've actually set up crystallization boxes that we ship to teachers. We set up a little bag of disposable pipettes and just cover slips and everything. We ship them off to the schools, and they set up trays there.
Have you ever tracked any of them if any of them end up in grad school or even?
I haven't actually. We have not, but that's something that we may be able to get information on.
Is that strictly a UF program that does that, or?
I don't know if anybody else does it. I know our one we do. The one that we do is a UF program, and we get funding for it from the National Science Foundation. I think maybe NIH as well.
But that's what I was wondering. Is
that Mary Jo Caroli?
Yeah, Mary Jo Caroli.
Yeah, this is exactly. a faculty member at UF who is just totally dedicated to this.
Yeah, to teachers. She just brought in a group of 80 students funded by. Again, she got funding for them to spend, I think it was maybe 4 weeks, just learning about infectious diseases. That was this past few weeks, and I gave them a talk on pathogenic viruses. And actually, one of the students came to see me just before I came here. She was really excited about science, and she lives in the Philadelphia area. So if anybody lives in the Philadelphia area who wants to take on a really excited undergrad to do research, let me know. But she was very excited. She wanted to know if I can help her find a lab over there and whether or not she could come back to the lab next summer to do research on viruses. So that's pretty exciting.
No shortage of labs in Philly. Somebody ought to be able to step.
Yeah. Step
Anybody
up.
from Philly? Yeah.
I would love to do an informal survey based on those remarks. How many people here in the audience were inspired to get involved in science while they were in high school? Raise your hand.
Cool.
Check it out.
Wow.
So it actually works. How about grade school? Nice. How about at home? Nice. All right. How about a TWiV? No, not TWiV.
Someday. We need time.
No, I think we've had a lot of people write us to tell us that our show has inspired them to get involved in virology. So I think that's
Well, you need multiple approaches, right? But the younger, the better, always.
absolutely.
Yeah.
So we're contributing to the glut on the employment market.
Yeah.
That's right.
No.
Exactly.
All right, I'm pausing because I'm told I asked too many questions, so I'm going to let someone else.
Not really.
See that? That's why I asked so many questions. So, Mavis, so now you like viruses, right?
Oh yeah, I like viruses and pathogenic and non-pathogenic. So we work. So another arm of my lab actually does a lot with emerging parvoviruses. Just trying to understand. Obviously, first of all, we get their structures, and then trying to understand. There's tropism determinants. What makes them pathogenic and while others are not. And we haven't really come up with a good answer at this point, other than maybe the toxic non-structural proteins that they produce. But yeah, so very interested in viruses and have been doing that now.
How do you decide what to do next, or is that not an issue? There's always plenty to do.
There's always something that comes up. We get data, that leads to new data. We've got some really exciting work going on with just the viruses. We're finding all these hidden functions in these very simple capsids. We've observed that the capsid, as I showed, cleaves itself with pH. So the question is why? So that's led to a whole new area of research. We've got an R01 to look at it. But not only does it cleave itself, it actually acts on an external substrate. So what substrate? Where? So now we need to track the virus. So really love seeing some of the talks where they're tracking viruses and the proximity ligation assay. I'm really waiting to do that. I talked to Becky about doing it to figure out what the virus is talking to on its way in. So there's always new things to do.
So in your talk, you had a section on trying to make antibody escape mutants.
Yes.
And let me see if I can do this. The idea was to construct a virus that, when you use it as a gene therapy vector, won't be trashed by the existing immunity in humans, right?
Yep.
So it seems to me that virus might nevertheless induce an immune response that's peculiar to that particular virus.
Yes.
Have you gone that far? Do you? I guess the idea is that as long as you can get the initial gene therapy in, you're a step ahead, right?
Yeah. So absolutely, you're correct, and we are very aware of that. So what we really want to do is that obviously there's a large percentage of us that do have pre-existing immunity to AAV, but there are some that don't. So you could always use one of the parent viruses for treating those who do not have that pre-existing immunity. The first thing we want to do is really just expand how many people can get a gene therapy. And so the first step there is to create a new variant that not too different from the old one that will expand that patient cohort. Now, if you need a second administration, we're going to have to do this again. But what we're hoping is if we can, I call it my toolbox. If we can have a little box that has, the green, blue, red, yellow vector, and I treat you with the green the one time, but I'm ready with the yellow, assuming this could get approved, we're hoping that you wouldn't have seen the yellow. So if you did need a second administration, we can use that. We haven't got there yet.
Is it known that the pre-existing immunity is actually a problem?
Oh, it's a problem. Yeah, it is known.
So what's the seroprevalence of AAV in the population?
So it depends on the serotype. So for 2, it could be, so AAV2 is the one that everybody works on in the lab a lot. It's not the first discovered, that was 1. But there's 70% immune prevalence for AAV2.
And we don't care, right? It doesn't hurt.
No, we've had it and, we weren't bothered by it, so we're fine.
Okay.
Yeah. So that's a problem.
And how many, how many serotypes do I have, Mavis?
A lot.
A lot.
A lot. Yeah. Not serotypes, genotypes.
Genotypes.
Yeah.
Okay.
There's over. So far, I was at a meeting at the American Society for Gene and Cell Therapy, and somebody said there's 800 different genotypes.
Wow.
Of AAV. Yeah. So they've been isolating a lot of them from people in China. So yeah, there's many of them out there, all just slightly different. So
this is a conundrum that you have a virus which is great for gene therapy, but we all have it already.
yep, absolutely.
So you can use your structural approaches to change, as Rich said, antibody sites. You can also change receptor attachment, right?
Yes. So that's right. So we know for the most of them, at least the primary receptor, a lot of them seems to engage a glycan on the way in, maybe kind of a sequestering interaction. So we know where those are. So we can try to change those either by, putting a peptide or engineering some sort of other recognition as well.
And the idea there is maybe you could target it to different tissues?
Absolutely. Yeah, that's it. That is the idea.
So I think one of the more advanced uses is for the blindness, right? You mentioned the other day.
Yes. And that's because the eye is considered an immune privilege site, but those are subretinal injections. So they stick the needle into your retina or subretinally and inject it. And I say I have an objection to a needle in the eye. I don't want it, right? So really, they're trying to put
But if
it,
you need the therapy, you can't see it coming.
That's right. But there's and so don't ask me about the anatomy of the eye, but there's a space called the intravitreal space, and that's where they really want to inject. But there is some immune response there that can be seen. So that's still a problem, but yeah.
So that has worked in clinical trials. It's not yet approved, right?
No. We don't. The U.S. does not have anything approved for gene therapy at this point.
Yeah. I wanted to ask you about this very cool paper you were on, where you took a germline sequence of a AAV and you predicted the structure of it. Is that right?
Yeah, that's right. So these were working with people in Australia, and they isolated a marsupial ancestral AAV. I don't remember how long they said it existed, a very, very long time. And yeah, so they isolated it and we predicted, so modeling, so we predicted the structure of it, and it looks very different. It's got some huge insertion loops. So what we're actually hoping to do now is solve the structure to just confirm.
So you have the sequence of the entire capsid protein, so you can produce it. And
yeah, absolutely.
Nice. I think that's great.
So that's
you can resurrect these old viruses. They're not circulating anymore, right?
No, they are not.
Is that useful for, I'm sorry, one more. Was that useful for
gene therapy?
Gene therapy?
Yeah. We think so. So in that particular case, it hasn't been tested. So we're going to have to test it against a panel of human serum samples, and also just ensure that it transduces or it could able to express genes in human cells. So we haven't done that test, but yeah, it would be great because in theory, we should have a low immune response to it.
So these vectors are strictly a gene delivery system. I mean, as long as you can package the DNA in the capsid, as long as the capsid gets in the cell, you're fine. Nothing else about the molecular biology of the virus matters for the actual gene delivery, right?
No, it shouldn't.
Okay.
It shouldn't.
So I'm just thinking if you got something from a marsupial going into a human, all you care about is that it's actually taken up by the humans.
Right, because for the gene therapy vectors, you don't actually deliver any of the viral genes, right? You're just using it as a vehicle. Right, it's a bag to carry your stuff in. So that's it. Exactly. Yeah.
I beat you, Alan. I beat you.
So there's a bunch of different gene therapy vectors out there. AAV seems to be one of really the most popular. What distinguishes AAV from others and makes it so good?
So I guess I have to then admit that it's less immunogenic than the others. So AD is very immunogenic, although AD has a much larger packaging capacity.
Yeah, I was going to ask you, what's the packaging capacity for AAV?
It's not great. It's about 5 kb. So we're trying to improve that right now. And then lentiviruses and retroviruses, I think there's still concern about integration and mostly. So I think that's why AAV is the most popular right now, even though it is the smallest packaging capacity around. Yeah.
Following up on Alan's question about computational biology being able to predict structures, and coupled with your structural biology knowledge, wouldn't it be possible to create something totally synthetic that doesn't exist in nature, would be a perfect vector for these genes?
Yeah, yes, definitely. And actually, a lab has tried to do that. Using they used the sequence of the AAVs available and kind of worked backwards to produce an ancestral AAV, and they call them ancestral AAVs. So it has been done, and then they were able to make it. They made it, and it does package virus. So these are ANK viruses by Luc Vandenberg.
Let me take a minute to tell you about the sponsor of this episode, Promega Corporation, which is locally here, right, the global headquarters right here in Madison, Wisconsin. For over 35 years, Promega has integrated the values of corporate responsibility and sustainable business practice into all aspects of its corporate culture and activity. They realize that success depends upon enduring connections forged among customers, community, and employees. Promega supports its customers by providing the highest quality products, the best possible customer service, and unsurpassed technical support. Their 400-acre campus here includes corporate headquarters, research and development, and full manufacturing facilities for life science researchers. Promega makes systems for extraction and purification of DNA, RNA, and protein from cells and tissues, and their systems integrate seamlessly, providing an uninterrupted workflow of products that help researchers develop the solutions of the future for diagnosing, treating disease, or discovery of new therapeutics. They hold significant intellectual property rights and licenses in several key areas that form a foundation for its diverse portfolio, including bioluminescence, like luciferases, luciferase reporter vectors, and substrates, short tandem repeat detection for STR-based cell line authentication, human identification, cell and tissue characterization, and mixed sample detection, halo tag protein labeling and capture technology. They have over 30 years of experience delivering reagents to life science researchers, and in addition to their standard catalog products, they also contract manufactured products or components for other IVD or medical device manufacturers, like clinical sample concentrator kits for purifying DNA and RNA, components for lab-developed IVD tests, and amplification reagents and master mixes for molecular diagnostics. Promega manufacturing meets or exceeds quality standard requirements to ensure on-time delivery of high-quality custom, bulk, or specially designed products. For more information about Promega, visit promega.com/ASV. All right, so Mavis, you, as far as I could tell, you have 170 papers, and I want to ask you a tough question. Which is the coolest?
Oh.
Assume nobody's here that's on any of those pages.
It's just us, Tom.
Yeah, which is the coolest paper? This is a tough one. For me, I think that at least where I am now in my career, the most important one was our structure of adeno-associated virus serotype 4. I think that kind of put us in the field. So maybe not necessarily the coolest, but that's when we first defined all those variable regions that I spoke about, and that's really driving a lot of the work that we've been doing. So, yeah, I would say.
Similar question as maybe the answer is the same, but as you look back, and is there a moment in your career that to you stands out as like one of the most exciting?
Well, that would have to go all the way back to a postdoc and solving my first structure, seeing the map. That is the coolest, right? When you, at least for a structure biologist, when you solve your first structure and you are able to build your model into it, that would be the most exciting.
And what was that structure?
That was the structure of feline panleukopenia virus that I solved. And I could tell you that even now, still get excited. I was saying to somebody the other day, we've been solving all these cryostructures, and the first time we got our cryostructure where we could see our side chain, Rob and I said, we're going to get popcorn, sit down, and look at our structure. and that's a true story. That's how much fun it is, yes. And we did.
I've often thought about people who do biochemistry on on a system or a protein for a long, long period of time and must create some sort of image of what this is like in their head and then get the structure, it must be absolutely mind-boggling.
It is. Yeah, I mean, it's just so exciting, yeah.
So do you think the field of structural biology, both X-ray and cryo, is it reached its maturity, or is it going to still get better?
I think it's going to get better. I think that the technology developments are going to allow us to actually now answer, questions about biology. So we get accused a lot of the time of, we're looking at isolated proteins and molecules. Now we could actually go into the cell and get the resolution we need to understand the interactions in the cell. So yeah, I think it's going to get better. I think it's going to really help us answer questions in biology a lot better. Yeah.
Of course, we wouldn't answer the opposite, so.
No, I would be out of a job.
A lot of people out here want to be scientists. What would you tell them? What advice do you have for them?
They want to be scientists.
They want to have their own labs and...
Right. I mean,
what advice do you have?
For those of you who know me, obviously science is fun. And I say if you wake up every day wanting to go into the lab and you're excited about it, then you're a scientist, and that's what you should be. So that's what I do. You could ask my students. I think it was my birthday, or I'm not sure if it was Boss's Day. Was it last year? Yeah, she's laughing. And so what my lab did is they just bombarded me with all these emails about data. All of them decided and made me, I think, a little poster of data. And that's because they know if they emailed me 11 o'clock at night or 12 o'clock at night with data, I'm going to respond. And I think for me, that's what science is about. I'm very excited, and I hope my students come out same thing, and just curiosity for data and excited when you see it. So I would say that's what you want to be.
So your limit is midnight? That's as late we can go?
Yeah, I normally have to email and say, I have to go to bed now, and then I say good night. So, but yeah, that's the way we are.
Alan?
No,
You're okay?
Good.
Kathy, you have any questions?
I'm good.
You're good? I have a few more. If you had not been a scientist, what would you have done with your life?
If I was not a scientist, I would have been a jewelry shop owner.
Jewelry shop?
Yeah, and that's because, and I've always said this, is that I would travel around the world and I would buy exotic jewelry. I have to be rich though, because I wouldn't be able to make money out of it. So, but I would travel around the world and I would buy exotic jewelry and I would have a little store somewhere and I would sell them.
Would they be tiny crystals on these jewelries?
Well, probably not, because I would go to like local markets and handmade. I would go to people. The locals make them. I'm not going to buy them from big jewelers. Just from local people making jewelry. So probably no crystals, but really fun-looking jewelry.
Do you have any virus-like jewelry now? Virus necklaces?
I do not. I have 3D prints, but I don't see. Okay, Nikia, now you know what to get me.
I think I have at least 4 virus necklaces.
You do?
Yes.
I do not have one. Were they handmade by someone, or?
Yeah.
Yeah, exactly. So there you go. There's probably a market for if you want to do that. So, yeah.
Mavis, what do you think is the most important thing you've given to the field of science?
Right now, I would say our information on the antigenic epitopes of AAV, because I think a lot of people are now going to be able to modify their capsids based on this information. That would be, I think, so.
What's this lunch question?
I put that in there. That's your lunch question.
Oh.
My lunch.
Yeah, if you
go ahead, answer.
We get 2 people, right? If you could have lunch with any 2 people, living or dead.
Assuming that they came back to life in their prime.
And I suppose it could be 2 lunches. It'd be fun to do it together, though. Who would they be?
Who would it be? Probably Dorothy Hodgkins would be one of them, and then who would be the other one? I'm not sure.
Yeah, you can get back to us.
Yeah, I
Yeah,
can get back to you on this.
Explain what you think lunch with Dorothy Hodgkins would be like.
Well, I think it'd be, talking to somebody who was in science and the structural biology field so early when it was dominated by men, I think would be, just how she handled it would be fun to find out, right? Because, I think she handled it well. So she'll be one of them. I guess, this may sound corny, but I would say actually Michael Rossman. I would have lunch.
Isn't that nice?
He's a great character, and he's been, such a mentor. And so my love for science comes from him. Because those of you who see him in Lily Hall, he'd be running down the hallway, this little old man, and every morning, if you gave him data, he would be just as excited about it. So him. He's alive. So
Great.
The only difference is that now he's in Hockmeyer Hall.
Yeah, he is. The basement was very dreary, I'm telling you. So, yeah.
All right, time to do some picks before we wrap up this episode. So Mavis, on TWiV, we pick something every week, science-y, and we didn't warn you ahead of time, but we'll do ours, and if you can think of something, we're welcome to hear it. If not, it's fine. Let me start with you, Dixon. Do you have a pick for us?
I actually do. It's from Cosmos magazine, the latest issue, and it's called Mars as You've Never Seen It Before. And it's the reconnaissance orbiter at high definition with really cool music. And it's neat. It's really neat. The surface of Mars is so bizarre. I mean, it has almost nothing in common with the Earth.
It's almost like it's from Mars.
It's almost like it's, Alan, you're redundant at this moment, please. So yes, these pictures are astounding no matter where they're from. They almost look like they're made up by computer-generated imagery, but they're real. And that's what makes it so astounding. I got my love for this kind of stuff from my father, who in the old days when television was first coming out in these little round screens, there were lots of space programs out to Captain Midnight and all kinds of other, Flash Gordon. And, getting inspired at that level as a little kid and then seeing how your father reacted, I just so wish my father could be here now to see these images, because he always wondered what these things were like. And he imparted that wonder into me. So I share them with you.
Thank you. Kathy, what do you have?
Well, I have something. I went to two presentations, shows, whatever, last Sunday at the Ann Arbor Summer Festival. Those of you from Ann Arbor may have known that Dr. Kaboom was there. Has anyone ever seen Dr. Kaboom? Wow. Okay. If he comes to your place, you should go. These are shows primarily for children, but there were two science shows. One of them was Live Wire, and the other one was It's Just Rocket Science. And he was, I guess, must have been born in Germany, but he's an American citizen, served in the army. And so he goes around, at least the country, if not the world, doing these science performance shows to get people interested in science, particularly children. And so the link that we'll have is his website. It's spelled D-O-K-T-O-R because he's doctor from Germany, Dr. Kaboom. He said if you just Google Dr. Kaboom, you'll get some guy in Canada who does balloons. So that's not who you want. So, but the cool thing is he did all these great experiments on stage, brought kids up to do stuff. And if you go to his website, it really captures his mission statement captures what I liked about it the best. Oh, I have to put my glasses on. Okay. Science is for everyone, not just the guy in the lab coat or the girl who wins the science fair every year. Science is not hard, but it does take effort. That's not hard, that's just work, and that's just life. Every child is intelligent, creative, valuable, and should know that about themselves. And it was really meaningful because the first children that he brought up, he asked them if they were smart, and they said yes. And he said, that's the right attitude. Don't let anybody tell you you're not smart. But take a look at his website. He's got lots of videos, so if you can't catch a show, you can go see Dr. Kaboom online. Really
Thank you.
Doing science outreach in a great way.
Alan, what do you have?
Well, for those of you who are down in the dumps about how 2017 is going, I'd like to reassure you that in just under 2 months, the sun is going to disappear. And so I've picked a site. August 21st is the total solar eclipse that will be visible across a swath of the United States. There are total solar eclipses periodically, but they're often in the middle of the ocean or in places that are hard to get to for a lot of us. This one is going to go, the line of totality is going to go from Oregon on an arc across the country to South Carolina. So probably most people who are in North America are potentially within range of this. If you're outside the line of totality, you'll get a partial solar eclipse, which is kind of cool. But I'm really hoping to get into the actual shadow. So I picked a site, eclipse2017.org. I think it's just some guy who's really into eclipses who put this site up. I'm not advocating any of his products or anything. He sells some solar glasses that he says are rated for looking at eclipses, so that might be something. But it's got a lot of information about where to go and how to view the eclipse safely, and definitely something to put on your calendar.
All right. Rich, what do you have?
I went to my bookshelf for this one. This qualifies as science because it's kind of sort of science fiction. I mean, it's
science fiction.
You think it's science fiction?
Sure.
Okay. Sirens of Titan by Kurt Vonnegut. If you haven't read it, I recommend it. It's a hoot. And you will, if you read this book, not only be amused, but you will also discover the purpose of human civilization. Okay? So go for it.
Thank you. My pick is Research America. Anybody who knows about Research America? Not enough of you. You all need to go to this website. This is partly why the NIH budget went up $2 billion this year. It wasn't just Congress saying this is the right thing to do. It's because people lobby. And this is one of the most important lobbying organizations for medical and health research. And so they have a website which explains everything they do. Mary Woolley is the president of this organization. I heard her speak recently at ASM. She gave a great talk. And here's one thing I took away which you need to remember. If you meet someone on an airplane or in a bar or an elevator and they ask, what do you do? Here's what you tell them. And she told us this, and it's great. You tell them, I work for you. And that's the best way to get them engaged, because then they're going to say, really, what do you do for me? And there you go. And that's an in. So this website, in addition to all the other stuff, they have the results of many, many surveys that they have done about science, and you can use them. There's slides that you can use in your talks. Things like, over 80% of Americans can't name a living scientist, right? And here's a good one that I like. Who does the public want to get their science from? Guess what the answer is? Us, scientists. So anyway, if you want to give a presentation, it's really great for that. But most importantly, you should support them. I'd like you to support us, of course, but if you have to support only one, support Research America, because they make the research go forward. Really, really important. Mavis, you have anything you want to share with us?
I do not.
Okay. That is TWiV 448. You can find it at Apple Podcasts, microbe.tv/TWiV. And if you have a smartphone or a tablet, there are lots of apps you can use to listen to podcasts. Please subscribe. It really helps us to have lots of subscribers. Even if you don't listen, just subscribe. Okay? It helps us to get more advertising.
Just tell Alexa to download the episode and play it for your dog.
And if you have questions or comments, we always love getting them. TWiV at microbe.tv. If you like what we do, consider supporting us. We have a page at microbe.tv/contribute where we have a Patreon account where you can give $1 a month, or PayPal, or you can buy t-shirts at cafepress.com/TWiV. I want to thank everyone for joining me today. LivingRiver.org, Dixon Despommier. Thank you, Dixon.
You're welcome.
Who's next? Rich Condit is an emeritus professor at the University of Florida, Gainesville, currently residing in Austin, Texas. Thank you, Rich.
Thank you. It's always a good time, and thanks to everybody. I'm so grateful for this.
Hear, hear.
It's great.
Alan Dove is at alandove.com. You can also find him on Twitter. Thank you, Alan.
Thank you. It's always a pleasure, especially with a crowd.
Kathy Spindler is at the University of Michigan in Ann Arbor. Thank you, Kathy.
Thanks. This is a lot of fun.
I'm Vincent Racaniello. You can find me at virology.ws. This episode of TWiV was sponsored by Promega. Promega's products are used by life scientists seeking to solve fundamental questions about biology. Promega supports researchers who are applying scientific knowledge to diagnose, treat diseases, or discover new therapies. For more information, go to promega.com/ASV. I want to thank the American Society for Virology. I forgot Mavis. I'm so sorry. Mavis, thank you so much for joining us today. Hope you enjoyed it.
Yeah, it's been a pleasure. Thank you.
University of Florida at Gainesville. I want to thank the American Society. Good thing I look at my co-hosts. But online, I can't do that. That's when
Yeah, I would have yelled at you. It's okay.
I want to thank ASV for having us. The organizers, Kristen Bernard and Paul Friesen, thank you so much for helping us. I want to thank Jay, the sound guy, who made flawless sound here, because it's not easy to make really good sound. And I want to thank the audience. So, to thank you, I'm going to throw out some T-shirts.
Knot 'em all up, boys.
His shoulder's gonna hurt.
Oh, you don't trust us to throw them now. That's
Nice. One more.
one more.
Now pitching for TWiV.
You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.